listening to his presentation at the Imperiled Butterfly Working Group, which is a organization. It's not an organization; it's a affiliation of basically uh, 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 state and local park managers, as well as academicians and regulators. We meet episodically, and the last meeting, Jaret gave a presentation that was very interesting. It made me recall that we have not had him as a presenter, and that's shame on us because, as previously noted, he is the leading butterfly expert in Florida. And so, with that, Jaret, I'll turn it over to you, and we very much look forward to your presentation.
Thanks so much, Dennis, and thank you guys for having me this morning. Always a pleasure to be here and talk to the Miami Blue chapter. You guys do wonderful work. I've known Dennis and Jason for quite some time, and many others here today. So really, again, my pleasure to be here, and thank you so much for that wonderful introduction. So this morning, this afternoon, I'd like to kind of provide a 10,000-mile view of the work that my lab has done in collaboration with a lot of different stakeholders, partners, individuals. I want to emphasize that I think we all know that conservation work doesn't happen in a vacuum. It takes the dedication of, many experts out there, many dedicated people, many volunteers. So just want to emphasize the work and the volunteer efforts that many of you have also provided to many of these South Florida taxa. It's really wonderful to have fantastic partners like the Miami Blue chapter of NABA. So I want to just dive in and talk a little bit about the last 10 years of work that we've done at the University of Florida. So if you dial the time scale back a little bit, roughly around 2017, 2018, we received funding from the Disney Conservation Fund. It's an unusual partner in many ways. Most folks don't realize Disney has a long-term sort of nonprofit arm that is dedicated to conservation giving. But historically, they've given mostly small kind of capstone awards of 25 to 35,000 in supporting work all around the globe. And just a few years ago was their 25th anniversary of giving. But they realized when they sort of delved into the projects that they've supported that they could do more. And so they launched an initiative that was originally called Reverse the Decline. It changed its name to Saving Wildlife, and they invested in 10 programs around the globe. And they did something really unique for a nonprofit. And then they realized that conservation is a marathon, not a sprint. And they know that it takes more than 1 year to recover an organism or to make progress on that recovery. So this initiative was a decade-long initiative. And we were very fortunate to be one of 10 global recipients to receive this award. And it was fairly deep funding, roughly $250,000 to $350,000 per year for the duration of the 10 years. And we're just winding up our 10th year this year in 2023, 2024. And Disney also had good vision in the fact that they also realized that good conservation work requires sound strategic planning. So they mandated that all awardees spend the first year of the award working with stakeholders to really craft out detailed strategies, kind of an architecture to ensure that they were moving in the right direction, that they had done their homework, and that they were focusing and ultimately recording metrics of success as they went along. So right or wrong, maybe I was a little too ambitious, but we ended up the day focusing on 42 different taxa. And this runs the gamut from those like the monarch, which are declining, to many that are critically endangered. And you will see several on this image from Shaus's swallowtail, Miami Blue, Bartram scrub hairstreak, Eumaeus atala, and then several Western species like Lang's metal mark, bay checker spot, and the list goes on and on. So we focused in two primary areas, California and Florida. not because that's where Disney does business, but those are the epicenters of imperilment within North America predominantly, because they share many things in common. They're large states that have a lot of endemism. They have a lot of specialized habitats, and they're under extreme population pressure in many areas, which means that they harbor many small populations that remain under threat due to increased human development, climate change, a variety of other drivers and stressors. And so this image just kind of shows some of the different project partners and locales that we work on, from the Florida Keys to the northern portions of California to San Bruno Mountain overlooking the San Francisco Bay Area. So it's some really specialized, really gorgeous, wonderful habitats, and we work with a lot of different partners to make this work happen. And as part of this award, we both do the work ourselves, as well as sort of strategically farm out work to partners. So the money that comes to the University of Florida, some of it stays here, some of it goes out and is subcontracted with partners across North America. And we focus on an adaptive management strategy, all kind of focused, centered around recovery. So many of the projects that we delve into are basic and applied research, habitat restoration and management, direct species management and restoration, monitoring, and also outreach to the public, because this, as you all know, is critical. If people are going to understand why these organisms are important, if they need to vote, if they need to be advocates, they need to understand that these organisms occur in their backyards and why they're valuable and what we are actually at risk of losing on this planet. And my lab has sort of divided this effort into a number of sort of larger thematic areas. And so one of them, which is kind of near and dear to my heart, is the development of best ex situ practices. We do a lot of captive breeding in my lab. These are all images of Miami blue butterflies being bred in my lab at the University of Florida. And the goal with this work is not just to produce organisms for reintroduction, but the broader goals of captive breeding are filling in data gaps of information, using organisms for research, producing organisms for reintroduction back into the wild, and also just to maintain populations in safeguard in case something happens to the extant wild populations. And so where possible, we also try to publish and to try to kind of expand on the knowledge and the techniques that we use that could be applied to other organisms, whether they're a blue, a hair streak, an emphalid, the list goes on and on. And we work with a number of different partners across the country that have similar ex situ efforts going on. These are images of Schaus's swallowtail. So it's fortuitous to us that some of these organisms are relatively very adaptable to captivity. Some are not. And so a lot of this work is pioneering because nobody has done this before in kind of large scale production. So the two that we focus on a lot are, of course, are Schaus's and Miami blue, for good reason. We have contracts through U.S. Fish and Wildlife Service, other partners, as well as funding from Disney to do this work. And they are arguably two of the most charismatic and most in need species in Florida. And so just to kind of highlight the productivity of our lab, since 2017, we produced over 35,000 organisms in captivity that go towards colony maintenance, restoration, and organism research. And so we are arguably the largest lab and most productive lab when it comes to ex situ work in the nation, which gives me a lot of pride because it's a lot of effort. We could not do this work without fantastic partners, and my lab is absolutely wonderful. So they make me look very good. And I want to emphasize, as all of you know, that this type of work is not something that you can start and then go away for a 2-week vacation or go home on weekends. It's 24/7, 365 days a year to maintain organisms. So it's a lot of dedication, a lot of hard work, but it's something that I take very strong pride in doing this work. And then we also use a lot of these organisms as well as surrogates to help understand the factors driving reintroduction success. So the images here that you see are with the Miami blue, and this was part of a 5-year U.S. Fish and Wildlife Conservation Recovery Initiative grant, which was meant to sort of test reintroduction strategies in the field to understand what worked and what didn't, because the field of organism restoration or reintroduction is a burgeoning field. It's mainly been dominated by vertebrates, birds, reptiles, and mammals, not so much insects. And in the past, a lot of the work that has been done has not been done, let's say, in a scientifically sound manner. It's like bringing individuals and releasing them back into an environment. If it works, we all pat ourselves on the back saying, great job. If it doesn't work, we say, that's too bad, but we don't know the reasons why. And so this project was trying to understand and disentangle some of the factors that drove success and or failure. And so we tested different things like the different ways of reintroducing organisms in the field. These are two little examples. One is a small PVC tube that has 1 or 2 Miami blue pupae in them. Others are larger that hang from trees that can have hundreds of pupae in them. We tested numbers at release, we tested life stage, and we tested both soft and hard releases. And so a soft release is one that organisms are just released back into the environment, and a hard release, or sorry, a soft release is one where organisms are reintroduced in the environment with some sort of predator exclusion, such as these large field cages that you see in the upper left-hand side. And this is Taylor Hunt deploying them at one of the South Florida parks. And a hard release is just individuals released back into the environment without any type of predator exclusion. So we did very detailed trials to better understand what would work, what would fail. And I think we got to some really good best practices moving forward. And one of the most successful was releasing pupae. And this in many ways makes sense because the individuals emerge, they're within that natal patch, it's kind of minimally invasive in many ways, and it can be deployed in such a way that also allows us to go back and really record success and failure. You can easily see from a pupa, did it emerge? Was it predated? Did it die? And so there's a lot of good metrics to be taken from such efforts. And this particular contraption, which we kind of dubbed in the lab little spaceships, could be hung from woody vegetation in the field. They could have and house over 200 Miami blue pupae. They have mesh at the bottom to allow rainwater to follow through, and they have a larger mesh at the top, so as the adult butterflies emerge, they can escape on their own without any human need for intervention. And then they're hung on filament, so even ants or other arthropod predators cannot predate those. So it's essentially safe from birds, herps, as well as any arthropod predators. And this was by far the most successful contraption. And here you can see 2 little adult Miami blues just getting ready to crawl out of the top of this little spaceship. And this one is pictured hanging at Bahia Honda State Park. So that has enabled us to move forward with more detailed reintroduction work at Long Key State Park, at Bahia Honda State Park, and increasingly at a few other locations in South Florida. So we have produced and released almost 15,000 individuals at South Florida conservation properties. That's the good news. The bad news, I guess, is while there was a population established in 2022 at Bahia Honda State Park, we no longer see evidence of continued success of that population. So again, just to emphasize, conservation and recovery work is a marathon, not a sprint. So we continue to work on this, but we have gained a lot of information over the last several years and a lot of detailed research to go and better inform and strategically look at ways to increase success in the future. And then if we segue to Shelse's swallowtail, you all know, the past story of this, and Jason's been involved in research on this butterfly as well. But this is truly a success story where we've come from in the past and the volatility of this butterfly to a low of 4 individuals in 2012 to numbers in the last several years eclipsing the highest numbers recorded in the wild since modern monitoring has happened. So this is arguably a really positive story. We hope the trajectory will continue. We will continue to work on captive breeding and organism reintroduction. And of course, we're trying to expand with the help of Florida DEP and others, NABA, to expand to other areas in the southernmost portion of the range, such as Lignavite Key Botanical State Park. And there were individuals recorded this past year. So we're optimistic that with continued reintroductions, we can expand the sort of string of populations south from northern Key Largo and enable this butterfly to exist in much more of a metapopulation structure as it historically probably did. So this again is a very positive story. And if you have not seen this already, if you view on National Geographic or you subscribe to Disney Plus, our work with Shasta Swallowtail was highlighted as the terrestrial example for America's national parks for the highlight of Biscayne National Park. And so you can see some of our efforts there. It's a wonderfully produced series anyways, with a lot of spectacular visuals overall. And then we worked with partners such as Fairchild Tropical Botanical Garden because knowing where to put organisms is half the battle. And we did a year-long extensive evaluation of potential reintroduction locations, targeting over 27 sites, looking at both biological criteria as well as logistical criteria, because access to locations, communication, or willingness of landowners to participate, these can be significant impediments. So we mapped the areas of host plant, we documented adult nectar sources, we rank sites, so we have a pretty good matrix of potential sites to expand to in the future if we're able to do that in the future. And this is going to be published in Restoration Ecology as a best practice document for other imperiled Lepidoptera. And then we understand that a lot of the work that we do, we need to get additional information, such as the genetic health of populations, understanding connectivity of populations, is essentially reconstructing the genetic population structure of these populations to help inform conservation and management actions moving forward. But many of these populations are incredibly small, they're incredibly sensitive, and so sacrificing individuals is really not an option. So we try to look at new non-invasive ways of sampling tissue material for genetic analysis. And this example is with the frosted elephant, a butterfly in northern Florida, a butterfly that's currently being evaluated for listing under the U.S. endangered species list. And so we worked on this project as a test example to see if we could use environmental DNA, essentially DNA captured from hatched eggs in the field, a way that would not necessitate capturing any individual organisms, removing tissue material from them, putting them in harm's way, and also in a way that could be deployed to community scientists so that we could expand the number of eyes and participants in the field to get genetic samples, because this butterfly occurs throughout many populations across much of Eastern North America. So certainly extends beyond the possibilities of our lab going out and sampling. And so this really was a success story. We could demonstrate that we could get viable genetic samples from hatched eggs, and we could reliably develop techniques that would enable community scientists to easily collect samples in the field without having to refrigerate material in freezers before they sent material back to us. So really developing new techniques that could be used in combination with more traditional techniques, such as removing a leg from an organism. And in this context, we also expanded the work to just collect material in buffer solution, which enables it to be just held without any type of refrigerant and without the DNA degrading. So all of this is meant to kind of expand the opportunity to collect non-destructive sampling for population evaluation, and then bring that material back to the University of Florida for analysis ultimately. And so since that project started several years ago, we now have actually more than 9 projects that are working on the range-wide population genetic structure assessment. And so these are some of the organisms, Florida-based organisms that we are working with: the eastern beardgrass skipper, or the Orogo skipper, the frosted elfin, Klott's sawgrass skipper, or the sawgrass skipper, Duke skipper, Florida dusky wing, and also several non-lepidopterans such as the blue calamintha bee. And these are all organisms that are either extremely rare or coming up for potential evaluation for listing under the ESA. And we just recently published work on the Florida dusky wing, and this was a good model project to look at how the fragmentation of pine rockland areas in South Florida affects the distribution and the genetic health of populations. And the big takeaways here were really a little bit surprising in some respects, that there was still some genetic connection between the Key Deer National Wildlife Refuge and the South Florida mainland populations for this butterfly, likely through northern Key Largo. And the only caveat there is I will mention that when you do genetic work, it's not necessarily what's happening today. There's a latent kind of period of evaluation, right? So this connectivity could have happened 5 years ago, even 10 years ago, and it still shows a signature of that connection. But it's still really interesting that there is some evidence of connection from very disparate populations in South Florida. And also that many of these small populations harbor really unique or private alleles, meaning those populations are very different from one another. So that has lots of implications for management and conservation, including the opportunity to translocate individuals to expand and continue to conserve the genetic diversity that remains in these small, highly fragmented populations. And then we try to use emerging technology whenever possible. And this was a project that was extremely fun for me and from my lab. So we used artificial intelligence and remote sensing, essentially drone images, to monitor host plants along roadways for the monarch butterfly. And of course, we all know that the monarch is a declining taxon. I neglected to remove that IUCN red list because it is now removed from that. But we know that populations, even in Florida, have declined precipitously. The Florida populations alone are down about 80% over the last 30 years. So while not critically imperiled, it still is a butterfly that warrants conservation attention. And so we started this project by mapping milkweed populations along North Florida roadways from about Ocala to Jacksonville and west to Tallahassee, focusing on 2 key species of milkweed, Asclepias humistrata, which is pictured here, which is probably the most important most important deep sand milkweed for the return migration of the monarch, because it's emergent and vegetative when monarch butterflies are recolonizing the Florida Peninsula and the Deep South from Mexico each spring, as well as also butterfly weed, Asclepias tuberosa. And what we found is in all the roadways that we mapped, that roadway populations of milkweeds were actually pretty numerous. All the areas highlighted in blue have milkweed populations, and the focal areas highlighted in red on this map have ultra high density populations of those 2 focal milkweeds. So these would be areas that, if FDOT is going to change their management practices, such as the frequency of mowing or the timing of mowing, these would be really good target areas to target for monarch conservation. And all of this relates to the nationwide candidate conservation agreement for the monarch butterfly, which FDOT is a member. And this is a preemptive agreement that energy and transportation companies enter into in case the monarch is listed by the endangered species list at some point in the future. And they promise to manage a portion of their held lands in conservation for the monarch butterfly. And for us, the timing and frequency of mowing is particularly important, because if these populations of milkweed along the roads are going to be viable long-term, we have to ensure that they bloom, they're vegetatively available for monarchs to colonize, and that they actually have the ability to set seed so these populations do not become sinks over time, that they continue to grow and populate and maintain the numbers over time. And so we went out in the field for this next component of this, and we geo-referenced each individual milkweed plant along many of these target roadway areas. We looked at different size plants, from those just emerging from the ground to those that were in full seed and everywhere in between. And then we flew drones over the roadside areas, and as the drone flies, it takes individual images, and if you stitch those together, you can sort of see the overall images. And we flew at 4 different elevations, and here you can see Asclepias humistrata kind of pictured from 10 meters to 25 meters. You can easily see that as you go up in elevation, the image becomes a little blurry, but it's still there. And again, if you kind of feed this data through a deep learning model, which is not my field, and we'll say I still don't quite fully understand how all of this works, but essentially you feed this through computer models, and you keep validating the data until you come up with a reliable model that allows the computer to actually identify these individual milkweed species through the image analysis. And we also have fed in images from roadway cameras going from 35 to 60 miles per hour, because this may also be a very viable means of monitoring milkweed. But preliminary indications are that this is a really viable way of monitoring in the future without having boots on the ground, a very cost-effective way of monitoring populations through time that FDOT is quite interested in. And then we also have done a lot of work on habitat restoration, a little bit here in Florida, but a lot with our partner organizations in California and the Pacific Northwest. So if you start in the upper left-hand corner, these are lupin plants being propagated and are outplanted on San Bruno Mountain for the mission blue butterfly. The lower left-hand side is a grazing fire and mowing study for barren silver spot in Northern California. Obviously, the middle lower image is the Northern Florida Keys and restoration for Chauce's swallowtail. And then the lower right-hand picture is locustberry, a collaboration with Fairchild Tropical Botanical Garden and the Connect Protect program to outplant locustberry for the Florida dusky wing. So these are the focal organisms featured in this slide. And then this shows just some of the torchwood propagation that Fairchild Tropical Botanical Garden has been working with us on over the last several years. And we're working with Sean Christopherson and several people, Jeremy Dixon at Crocodile Lake and U.S. Fish and Wildlife Service, to identify locations where these plants can be outplanted, not only trying to boost the number of available resources for Chauce's swallowtail today, but look at future areas where the butterfly could either naturally expand to or be reintroduced to in 5 or 10 years in the future. And just to mention that these host plants, as you all know, are useful for Chauce's swallowtail, but also the Bahamian swallowtail, which is really still considered a fairly uncommon butterfly. And then the work with Fairchild Tropical Botanical Gardens, which is a great community effort, as you all know, with Connect Protect, and an opportunity to really understand how this butterfly ultimately colonizes kind of this urban core within Greater Miami and uses the Connect Protect network to kind of expand and move between existing pine rockland fragments. And some of the outreach material that we've done and developed in collaboration with Fairchild to help identify Florida dusky wing compared to other common dusky wings in South Florida. And then we've worked with FDOT in North Florida to revegetate retention basins. So FDOT has a myriad of different retention basins when they build a new roadway. Traditionally, these are waste areas that are not in the typical conservation vernacular, but they're large areas and they sort of exist like beads in a chain. So they offer a lot of opportunity for restoration, for rebuilding habitat, for connecting from one area to another, and also as migratory flyways for organisms like the monarch and other butterflies. So we've done work on dry basins and wet basins and outplanted a variety of different focal host plant species and nectar plants, both for butterflies as well as for all sorts of other native pollinators. And so these are just some images of some of those basins over time and the work that we've been able to do. And we go in and we seed, we compare plugs, and we compare containerized plants for what is the most cost effective, what actually is the best bang for the buck, and what will ultimately be most useful for establishing habitat for all of these. So we've outplanted to date probably over 15,000 plants in North Florida at a network of different retention basins. And FDOT has crafted these signs that they place out. So they've really embraced this as a viable conservation initiative. And many of these areas are colonized almost immediately by some of the focal organisms, such as the monarch. And then we try to, where possible, work on applied research, particularly for the green industry, because we have to realize that moving forward, in order to recapture habitat and to save biodiversity, it can no longer be just the wild conservation lands, but we need to work within the built environment, the habitat areas that you and I exist in every day. And so this is an effort trying to work with nursery growers, with landscape maintenance professionals, and with the general public to understand how we can develop, certify, and promote wildlife-friendly plants much more effectively for use. And so our goal with this is to really move the industry forward so that when you or I go to a big box store, we have a choice not only of an expanded network of wildlife-friendly plants, but we have some guarantee that when we buy that plant, like say we go and we buy a milkweed from Lowe's or Home Depot, we have to have some understanding that that plant has not been treated with systemic insecticides, that it's safe and viable for the organisms that we actually want to buy it for. And so we've been working using monarchs, milkweeds, and aphids as an integrated pest and pollinator management strategy for ornamental plants. It's sort of a model system that we can evaluate, looking at not only the chemical inputs, but also the effect of pest control opportunities, because ultimately growers have to have an option to battle soft-bodied insects like the oleander aphid, which is nefarious for colonizing these organisms. And so I won't go through this in detail because of time, but just to say that we've been evaluating the chemicals, and we've also been looking at how the aphids themselves could affect the viability of monarchs on these plants. So in this example, we looked at different aphid densities, and we compared monarch oviposition preference as well as the survivorship of monarch larvae. And what we found, surprisingly, is just the oleander aphids present in fairly high numbers on these plants is enough to significantly reduce monarch oviposition and ultimately the fitness of the larvae feeding on those plants. And this is likely due to several reasons. One is the fact that the cardenolides in the plant, the chemicals that the monarch larvae sequester, which render them toxic to vertebrate predators, is an inducible defense. So if you have another herbivore like the aphids feeding on that, the plant extends and expands the amount of chemical it produces to try to thwart that pest on the plant. Well, there's a limit to what monarch larvae can tolerate without having negative effects on it. So in this context, even just having high aphid load can render high larval mortality. Even the individuals that survive are lower weight and ultimately have a shorter forewing cord length, meaning they're smaller adults, which means ultimately they're less fit for migratory for a migratory life cycle. So all of this has real world impacts when we try to outfit our home gardens, our neighborhoods in conservation for organisms like the monarch. So this is continued work where we're going to be evaluating an additional array of different chemicals. We're looking for alternatives and best practices that we can push out to the green industry, to homeowners, to ensure that when you create a butterfly garden, it's going to be safe for the organisms, you're going to have better choices, and ultimately you're going to be more successful in what you want to do. And we also are trying to develop a variety of different educational materials for those different stakeholders as well, with the ultimate benefit that we want to create more viable, sustainable, wildlife-friendly landscapes for the organisms that we ultimately want in our backyards and neighborhoods. And then kind of winding down, you all know, of course, that we're at the Florida Museum of Natural History. And so engaging the public is really part of our mission, right? It's part of what we feel is critical. And my lab really takes this to heart. So we work on a variety of different educational campaigns, from developing exhibits to outreach with groups such as Girl Scouts, homeschoolers, events. In the lower left-hand side was a release of Miami blue butterflies that just turned organically into a large public event of just visitors to Bahia Honda seeing what was happening. And you can see the number of interested parties looking at federally endangered Miami blue butterflies being released. So it really does show what we can do for the public. And then something that I have done for many years now in collaboration with the craft brewing industry, and something I feel has been very effective and very fun, is the development of beers for conservation. So over the last 6 years, we've released over 13 different specialized beers in support of butterfly and insect conservation. All the money that is raised through merchandise sales and a percentage of the beer sales funnel back to directly support these organisms. And over time, we've launched many different really kind of rich labels, and tried to, even on the label, provide information about the organism, so that even if all you do is drink a beer, hopefully you go home with understanding a little bit more about the organism, and that it alone can move the needle. And then we have public events where we do merchandise sales. We had a national campaign for Monarch beer. We've used augmented reality with labels, and we've even swabbed organisms and used the yeast from the organism themselves to brew the beer. So over time, this has been really successful, and we've now started to pair this with native plant giveaways, targeting kind of target plants related to the organism featured on the beer. And over the last year or so, we've given away almost 2,000 native plants to the general public, and this has been very successful. So in this context, in this image here, you can see the blue calumet, the bee, in the upper right-hand side being swabbed, and the yeast being used to brew the beer for this effort. And just some of the images from this. But every time we give away plants, we give them a little card that if they send back a picture of the plant planted in their yard, we'll send them a free admission to a museum fee-based exhibit or some other token. And then that gives us the opportunity to map all of these plants across a larger landscape. So it becomes like a gap analysis. We can understand where we're having really good impact. Are we reaching sort of, depressed areas of Gainesville? Are we reaching, only affluent neighborhoods? Where might be kind of synergies where we could build on other partnerships? And so this becomes an opportunity to sort of map wildlife corridors across an urban gradient. And it's something we're going to be expanding in 2024 through a series of events to continue. I think it's just a fantastic opportunity for good community will, and we get a lot of data from those types of initiatives. And just to show how this has also helped raise awareness, this is just one snippet from the media generated from my lab, and this was the initial beer release, press release that went out for the Monarch beer, and over 176 million potential views just from that one press release. So it generated attention globally. It generated attention significantly in North America, Mexico, and the US. And so it really does kind of push the needle when it comes to understanding the benefits. So, it builds collaboration, it builds broad public appeal, supports local businesses, increases funding, and ultimately all that money goes back to insect conservation. So over the last several years, just through the beer events, we've raised over $35,000 for conservation. And we just had our last event, which was a moth beer, highlighting the impacts of nocturnal light pollution, and this beer called Nightlife, which is still on tap at the First Magnitude Brewery in Gainesville. And then just in kind of sum all this up, over the last 10 years and working with Disney and many other partners, you can kind of see all the stakeholders mapped out here where we have work. So we have a broad network of fantastic partners, almost 72 different partners across the country for working on all those 42 different taxa. And through this effort, we've generated $3 million. We've leveraged that into 1.5 additional $1 million. We have 74 strategic partners. We work on 42 focal taxa. We've bred in captivity over 44,000 organisms. We've released in the wild with our partners over 48,000 organisms, and we've outplanted, and this number is actually quite low, but this is the estimate we have, over 30,000 plants for habitat restoration, and conducted just at the Florida Museum of Natural History over 170 public outreach events over the last 8 years. So we hope we're making progress. We hope we have moved the needle a little bit. And I just want to acknowledge all the wonderful partners, all the funding opportunities, my lab, and of course, all of you for your engagement, your dedication, your hard work in the field, and your unending support for the butterflies of Florida. Without partners like you, we would certainly not have the opportunity to be successful. So thank you all so much for the good work that you do, and I'll stop here and be happy to answer any questions that you may have.
Greetings. So you can either type in your questions in the chat or just turn on your mic and go speak. I'm pretty sure Barbara has a question. Do you want to start off?
Oh my goodness, you pointed me out. Give me a few moments. I'm digesting all of that incredible news, and I'm just wondering what happens now as you're coming to the end of these funds.
Yeah, it's a great question, Barbara, and something that's very on the back of our minds. So, we hope that their support will not end. And as you saw, we try to leverage resources to other funders. So one thing that I take pride in is kind of the diversity of funders that we have, from private donors to nonprofits to federal and state agencies. And so we have a good network of new projects coming on board for the next few years, mostly funded at the state and federal level. This includes actually bringing about 3 more species into captivity. So in 2024, we'll have about 5 species total in captivity. But it is a constant battle, right? Constant battle looking for funding. And I think Disney has realized that their investment has been hopefully very worthwhile to us, that they've gained a lot of traction out of it, that we've done a good job of leveraging their resources for additional resources. And so I hope that they will continue to fund us in some level moving forward. It certainly will not be at the same level, but we've grown over the last 10 years with a huge network of different partners and funders and collaborative projects. So I hope that we can continue to expand that network and to continue to be successful in writing and receiving grants and contracts. So that's always my positive outlook on things. But it is, as you know, a constant battle. And so that's my job as the lab director and PI, is to search every possible avenue for funding.
Right. Do all the work and look for funding sources at the same time. Exactly. We have an interesting project coming up at Trek Agro Eco Park, and I'll be sharing information because they have just a bit very degraded pine rockland there, and we want to populate extensively with natives. So we want to get the native pollinator plants in that are from pine rockland. So that's going to be exciting, while at the same time we grow food so that we can showcase, yes, you can do both at the same time.
Yeah, that's great. And I didn't mention in the presentation, but all the work with the green industry efforts, the ultimate goal is to eventually develop a wildlife-friendly plant certification program where we can evaluate the chemical inputs and trial the plants to also better understand what groups of insect pollinators they're attracting, sort of akin to proven winners or some of the other larger plant trialing initiatives across the country. And Florida is an ideal location to do that, obviously. So the goal would be ultimately at some point in the future, if you go to a nursery, there could very well be some indication that this plant has been verified as wildlife safe and also, it's good for, bees or butterflies or birds or whatever the case is. And that's the ultimate goal, because there's so much discrepancy. If you go to a big box store, you often see plants that are mislabeled by species. You see plants that are aimed at, the monarch, but they're incredibly toxic still because they've been treated with systemic insecticides. So all of this provides tremendous public confusion, and we need to do something about it. Regarding the chat question, so iguanas are still at Bahia Honda. They're less of a problem. The Bahia Honda has actually come back quite well over the last several years, so the habitat looks really good. It is still a target location for restoration for the Miami blue that we hope to continue to focus on in 2024. Regarding Mary's question about, is there still enough genetic diversity to sustain viable populations within the Miami blue population? So that's a huge worry for us. And so one ongoing project that we have, which will be finished up in early 2024, is we've gone back through all the samples that we have from the original Miami blue population from Bahia Honda to contemporary individuals, going back in time through collections. So about a 20 to 30 year span of samples to understand if there's significant genetic erosion in those populations, if we've lost a lot of genetic diversity, and sort of what is the genetic status of the Miami blue currently, because it's probably or arguably one of the most, if not the most, imperiled butterfly in North America. You could put it up there with Lang's metalmark or a few others, but it's really down to incredibly remote, limited populations. And so genetic viability is a key issue, right? We're starting to see some worry of impacts in the laboratory population. So we'll have better understanding, hopefully by June of 2024, of what the genetic status of that butterfly actually is. And regarding where would I find complete life cycle photos of all the Shasta swallowtail Bahamian, so we actually do have some education materials I can forward to Miami Blue chapter that we've used for training, that has most of the larval instars pictured. I can probably add to that if you all wish that to be the case. Dennis, can you speak to your monthly Zoom presentations regarding research being done? Not sure I understand the question.
Dennis, can you use your microphone, please? He might be on the iPhone, though, I think. I'm not sure. What about Mary Rose? Your mic is on. Did you have a question or a comment?
No.
Okay. Is there anybody else that wanted to
Jason, I'm here. Jaret, I didn't properly describe it. I recall getting on an email list, I think coming from your location, about monthly presentations.
Oh yeah. So
I listened to a very good one about monarch migration through Florida, and I thought it might be something to tell people about, and then we can get maybe posted on our website so people can listen. They're fascinating presentations. I think they're typically at noon and they last about an hour, and I'll shut up.
Yeah, no, so we have bi-weekly seminars at the McGuire Center. So I'll try to add all of you. I'll contact the person running those and to get you all added to that. They're all available in person or via Zoom, and we have folks from around the world attending, but we try to pick sort of emerging areas of Lepidoptera research. So I'll make sure that, Dennis, that you all get on that list.
Yeah, thanks. It's fast. Just for the rest of the group, it's fascinating. It's deep science, but the last one I listened to was about monarch migration through peninsular Florida into Mexico, which I had no idea, but it was fascinating.
Yeah, there's some indication that they can go through the Yucatan and the Florida population has been very poorly studied. Jason, your question about how long does it take the chemicals to go out of a plant for it to be safe for caterpillars to eat. And that's a hard question because we don't know. We don't know what chemicals were used during treatment, and we also don't know the rates that were used. And we do know that nurseries often use kind of a cocktail of chemicals. And so, and sometimes some nurseries, as you might imagine, they think, well, a little bit works well, so a lot will work even better. So it's really hard to go and kind of disentangle that question. So we're trying to develop ways in which we can engage the green industry growers to use new emerging chemistries that are less toxic, or develop sort of best practices for the plants that are ultimately going to be used as larval hosts. So they can still have some pest control efficacy, but it won't ultimately harm downstream the organism. And so some of that could be using different chemicals, using different label rates. Some of it could also be holding plants back just a little bit before they're available for public sale. So all these are areas that we're researching, because we have to give nursery growers something to utilize to fight these pest issues, but we ultimately want the plants to be safe and viable for a consumer public. Go ahead, Dennis.
Yeah, just one more comment. You mentioned the Disney efforts. If there's anything we can do, even if it's a letter of support, I know it may or may not make a difference. Sometimes it does, to hear from the locals, so to speak.
Sure.
Just let us know. We're happy to write a letter or
thank you very much.
Otherwise engage them to tell us what to say. And you know, too often, as you know, I have to send nasty letters to bureaucrats, but sometimes I get to send a positive letter, and we'd be happy to do that.
No, that's wonderful. Thank you. Barbara?
Yes, you said that you did a study on the aphids, and how high the catenoids were on the Mexican milkweed. But is the Mexican milkweed just in general also higher in those catenoids?
It is. It's high. It has one of the higher cardenolide loads of milkweeds, but it's arguably the most commercially available milkweed. I mean, so you know, we've had some criticism about using that model, but you know, unless there's an alternative that's really viable, we're not going to remove that plant from commercial production. That won't happen overnight. So we have to ensure that if you have a lay audience that's going and buying with the purpose of trying to do something positive for the monarch, that you don't set them up for failure, right? It has to be something that's going to provide positive opportunity for the public, but also doesn't harm the monarch in the process. Because you could imagine that if these chemicals reside for 6, 8, 12 weeks in a plant, that's a long time in a garden that it could be non-viable, and all that time larvae are not surviving. So these become real sink populations, and really nobody's really looking at the negative impact this potentially could have on target organisms. So we have to really hit it from both sides ultimately. But you know, the aphids affect all milkweed, some more than others, and you know, we also have to give homeowners some good indication of what to do with those milkweed, those aphid outbreaks when they experience them. Yeah, exactly. But not all homeowners are as diligent as you are going to be willing to do that. They're going to break out the nuclear weapons, and you know, we don't want that. So it's a challenge.
Yes. And one other question. We had a speaker from University of Georgia, the Bulldogs, that have done the work on OE, and he was just saying and we have it reported on our website, and I've been meaning to like connect you two together, because he was saying that we shouldn't be planting any milkweeds down here, that they're all going to be infested with OE, and likely that every monarch that we have flying around in our environment has OE, which was just devastating news. And he kind of wanted us to figure out a way to start taking some samples or capture. And of course, the question would be, how do you do that without harming the butterfly, but to just get a handle on how prevalent it is in the environment here. So
yeah, I mean, Sonia Altizer's lab at University of Georgia has, you know, Project Monarch Health, where they have a very simple protocol for sampling OE, and you know, it's present in all background populations in the monarch. I mean, you know, and certainly at higher outbreak levels, it can be impactful, but I'm still on the fence a little bit about does that mean that, you know, what does that ultimately mean? I think there's actually more things to worry about with the monarch necessarily than just that. It certainly contributes, but
Yeah.
it has to be in the fray, definitely. And
Right.
then
I had an experience,
well,
sorry.
All right, so we're going to actually conclude with Mary Jackson's statement or question in the chat. So
okay,
can you read it? It's about
sure.
actually, yeah.
Yeah, so she asked, a research group is engineering or re-engineering the mammoth using retrieved DNA. Sounds extreme, but could something similar be done for a butterfly? So yeah, so this is using technology like CRISPR, which is gene editing or genetic engineering technology. And there's a lot of interest potentially in de-extinctioning species. And this is being proposed for even some extinct butterflies. It is extreme. It is extreme. And I think I personally am not a fan because I think we shouldn't be trying to revive or de-extinction things. We should be putting our energy into ensuring that the next Xerxes blue doesn't happen. And because at some point, if we're successful with some of these efforts, I think we don't know all the dangers of this. We don't know the downstream effects, but it creates a scenario in which, well, we can just let this thing go extinct because we can bring it back. And I think that's a very bad model. I think we need to understand that biodiversity is sacrosanct, that we need to keep it. We need to do everything we can to prevent organisms from going extinct. And that's an extreme example that should only be used under very, very limited circumstances. So with that, I'll just say thank you all again. Pleasure to be here. Hope you all have a wonderful day. And again, just thank you for all the great work you guys do.
All right. Well, thank you, Dr. Daniels, for your presentation. Very informative and educational. And we look to hopefully have you again in the future for more quarterly meetings. All
Definitely.
right.
Bye, everybody.
Bye-bye.